Welcome students to this week's session of the Algorithmic Thinking in Bioinformatics course. We are going to look at this particular question on which DNA sequences and motifs are bound by regulatory proteins such as transcription factors. If you feel a sense of deja vu, it is very normal, I am with you because we are seeing this particular biological question for the third time, for the third week, right. Initially we looked at this particular question of motif finding in the context of randomized algorithms and Gibbs sampling based algorithms, right. Then we looked at it in the context of EM algorithm. So, EM algorithm could be applied to find motifs in sequences, and now we are looking at it for the third time, but do not despair. Even though the question is old, there is a twist and turn, there is a fresh twist here. The solution is going to be different. We are going to offer a deep learning solution. And why is it that we are interested in this? What motivates us to study this particular deep learning solution to this problem? This particular deep learning solution is based on a paper called DeepBind. It was a very well written paper, and it was one of the very first successful applications of deep learning in bioinformatics, particularly to the problem of motif finding. And then at that particular time it was released, it was able to unify motif finding across a wide array of data sets, including data sets on DNA binding proteins like transcription factors as well as RNA binding proteins, okay, and coming from very different technologies as well, ranging from CellX, which is one technology, to ChIP-seq, to CLIP-seq and so on, okay. And so this is a very unifying model that was proposed and a well written model and one of the early applications of deep learning in bioinformatics. And so it is a good model to learn, so that the skills that you learn from understanding this model can be applied elsewhere, both in bioinformatics and beyond. It is a more self-contained introduction to a deep learning model as well. Okay, so with all these motivations, I hope you are now enthusiastic to learn more about this DeepBind model and this deep learning solution. And then we are going to follow the same systematic style that we have followed in all the other lectures. So, we are going to start with this biological question very briefly, because we have already seen this question before. How do we find the DNA sequences as well as the sites or motifs within the sequences that a regulatory protein like transcription factor binds to, okay. So, there is this twist here that we are not only interested in the motifs, but given a new sequence we want to also test if the sequence will bind to the protein or not. And what is the associated computational problem? Here is the new approach. We are going to take a machine learning paradigm, a supervised machine learning paradigm. By supervised, we mean that we have a training data where we have certain sequences which are known to bind to the protein and certain other sequences which are known not to bind to the protein, right. So, these are these positive examples and negative examples, and then using those two sets of examples, we are going to predict if a new sequence comes in, we are going to predict if that sequence will bind or not bind to the protein of interest. Now, we can also frame it as not as a two class prediction problem, but as a continuous score prediction. If you have certain data sets where you have binding affinities or binding scores, right, the strength of binding of sequences, different sequences to the same protein, then you can also learn a model for the protein that given a new sequence will predict the binding score or binding affinity, okay. So, those are the two problems. And the supervision comes from the labels that are assigned to the sequences, whether a sequence binds or does not bind, and then in terms of what the score of a sequence is in terms of its binding to a protein, okay. The algorithmic solution is going to be this DeepBind model that I referred to. So, this is a deep learning model, and so we will have certain standard algorithms that are adapted to this particular DeepBind model for training the model given this training data of positive or negative sequences or the sequences with binding scores, testing the model on unseen sequences, and then interpreting, more importantly interpreting the model so that we can understand what are the motifs that the model has learned, and then what is the effect of causing mutations that are associated with diseases on these motifs, okay. So, that is the plan here, and let us just very quickly look at what the problem is. So, you may have a DNA binding protein such as a transcription factor that can regulate the expression of many target genes. We have already seen there are these clock genes which control the circadian rhythm, right. In plants it is called CCA1, and then there are many genes that biologists have figured out that are target genes that are controlled by CCA1, okay. And then we wanted to know, at that particular time we were interested only in the motif finding problem. We will just expand the problem scope slightly so that we can also convert it into a sequence prediction problem or binding prediction problem, but at least focusing on what we had seen before, the motif finding is given the set of gene sequences and the upstream regions, and we know that CCA1 binds to the upstream regions, these blue regions of these genes. We want to find these hidden messages, right, these motifs that are implanted within these upstream regions of these genes where this particular transcription factor binds to. Okay. Here the hidden message, these A's followed by a TCT is implanted with a few errors in there, and then that is shown here where exactly it is implanted. But of course, when we look at real world data or you do not know the locations, given just the sequences which are shown here, we need to figure out what is the motif that are hiding within these sequences, right, and that is what is summarized here. So, where exactly does the transcription factor bind upstream of the target genes, and in this particular motif finding problems, you are given a set of sequences and trying to find these patterns. You can take two approaches. Experimental motif finding is going to be reliant on experiments like ChIP-seq, ChIP-seq and PBM. So, chromatin immunoprecipitation sequencing, PBM stands for protein binding microarrays. So, basically these are different techniques that will tell you which proteins and which sequences bind to each other. Okay. You can design it for certain proteins and then it will tell you the binding preferences of these proteins in terms of which sequences they like to bind to, which sequences they do not like to bind to, and then they may also give you scores of binding to these sequences. Okay. But then this is still not sufficient. The experimental space is huge, okay, and it needs computational approaches to zoom into the specific motifs within the sequences that are returned by these experiments. These experiments may return still a huge or not huge, I guess, reasonable size sequences. ChIP-seq may return sequences of length 100, okay, and the motif may only be of length 15 to 20, for instance, right. And so, figuring out which 15 mer or 20 mer within this 100 length sequence, right, that is returned by ChIP-seq contains the actual motif where the transcription factor binds to is an additional computational problem. And so, these experiments give you certain results that make it easier to find the motifs, but then we still need computational approaches to refine our results. Computational motif finding, on the other hand, given a set of upstream DNA sequences, it finds the exact locations of the motifs, right, where this transcription factor will bind to. But then if you have very long sequences or very short motifs, right, so very long upstream sequences of genes and very short motifs, then this is a needle in the haystack problem, right. It will be very hard to figure out where the motifs are. So, you need some form of handshake between these experimental and computational approaches. The experimental approaches should restrict the region of the upstream sequences that we are interested in looking at, and then the computational approaches then should help to narrow in within those and then find out the exact particular site within those shorter sequences where the motif is likely to be in. Okay. So, we need a combination of experimental and computational approaches, and this deep learning solution that we have actually tries to combine these two approaches in a very nice manner. Because the deep learning approach we are going to look at today is going to be applying computational analysis on a set of experimental results coming from experiments such as PBM and ChIP-seq. It is a good idea to briefly look at what PBM and ChIP-seq are. Okay. PBM stands for protein binding microarrays, and then ChIP-seq stands for chromatin immunoprecipitation sequencing. So, PBM is an in vitro approach to detect protein DNA binding events, and ChIP-seq is an in vivo approach to detect protein DNA binding events. So, what do we mean by that? In vitro is in a dish. So, you have a protein, let us say a transcription factor of interest, you want to know all the different DNA sequences that it likes to bind to, and then the DNA sequences it does not like to bind to. Okay. And the way that is figured out is that you have a microarray which is this little flat plate, glass plate, let us say, and then you have these different DNA fragments that are printed onto the microarray. Okay. Maybe let us say all possible 4 mers or all possible 6 mers and so on, okay, that are all printed onto this film, and then you then expose that to the protein of interest or the transcription factor of interest, and then the protein will bind to certain DNA fragments that it likes to bind to, because proteins do not just bind randomly to all the fragments, it has certain affinity, right. It only binds if it sees a certain pattern in the DNA, and so it just binds to those particular subset of DNA fragments. Then you can later look at the locations, you can just attach some fluorescent proteins and then look at which spots in the microarray light up, and using that you can figure out what are the DNA sequences that the protein likes to bind to and which are the DNA sequences that the protein does not like to bind to. Okay. All of this is done in a dish outside the organism, that is why it is called as in vitro. So, ChIP-seq is an in vivo approach where it is performed within the media where the cells are growing or the cell lines are growing, right. Let us say that you are interested in figuring out all the transcription factor binding sites of a particular transcription factor like P53, right. Within the cell, P53 has already bound to certain DNA regions. You just try to cross link those and then make sure that they are bound now permanently. You then fragment it from the rest of the cell and you use an antibody to pull down your P53 proteins along with the DNA that it is bound to. Okay. Now, these DNA are purified and then using sequencing you can figure out what are all the regions in the genome that this P53 or whatever transcription factor of interest binds to. Okay. So, regardless of which technology you use, you have a set of sequences, thousands and thousands of sequences, and for each sequence you have the binding status of whether the transcription factor binds or does not bind, and also a binding score or strength in certain experiments on the affinity of binding of that sequence to the transcription factor. Okay. Now, given this data as a training data, can we then solve the problem of interest? Okay. And before we go to our deep learning solution, it is good to remind ourselves of some early history of this motif finding problem. The reason that we keep on revisiting this problem the third time in this course is that I have said before that there is this paper on the EM algorithm and the rise of computational biology where they say that motif finding is a very fundamental problem in quantitative or statistical model of biological data, and then so in that sense it is a very central problem in bioinformatics, and so many approaches has been taken, the consensus or kind of string based approaches, probabilistic approaches using randomized or Gibbs sampling based algorithms, and then these recent approaches, and today we are going to look at this deep learning based approaches to motif finding. Okay. And this deep learning approach has an additional benefit. It enables not only motif finding, but can also make predictions for new sequences. Earlier methods can also do that, but somewhat indirectly. Here the deep learning method makes it as a central problem. Okay. But basically the central problem is predicting if a new sequence will bind to a transcription factor or not, and as part of that building that model, it also figures out what the motifs are for this particular transcription factor.